Thank you, and thank you, Nigel, for inviting me to this talk. As Nigel already mentioned, I have a kind of my affiliation is changed. So I'm based at South NIBSC, site of the MHRA, which most of you may know as NIBSC. And we still do the same thing, just changed our legal name. And today is my anniversary. I started there 13 years ago today. And so what I'm doing at the moment, I'm leading the vaccine group, and we do development and standardization of assays for evaluation of potency and immunogenicity of vaccines. And we do this in 2 ways. Majority of the people in my group are part of the national control testing for biologicals, in this case the vaccines. And myself, I'm more involved with the WHO and the development of international standards. And Nigel has kindly mentioned a couple of them already. So standards are important, but the important thing of a standard is the unitage you assign to that. And it's something that we do every day, as shown in these examples. And we're mainly familiar with the international system of measurements. Well, so it's important that we use the same unitage. And I have an example that I developed for students, but I'm going to give it here. So if you were offered 4 bags of gold, and each of them has an amount on written on the top, 8 kilo, 400 ounces, 1 stone, or 50 pounds. Which one will you choose? Now, I think if you're English, you can see I'm starting with the math. You may actually do the math. I'm Italian, we don't use anything other than grams and kilograms. So I have an app on my phone that I can have something. I can give you the answer. It will be the 400 ounce, and then if you want, you can do the calculation yourself. But so this is to highlight that if you have the same unit for everything, it's actually to make a decision to compare data between studies, for example. And this is particularly important for biological products. This is because they're quite complex in what they actually are. And the manufacturing of them goes through several steps, and although very well controlled in a GMP manner, there still is some inherent variability, and there is a batch variation. And what do you want to measure is not how much you have in that as a mass, but the biological effect. So you have to have an appropriate unitage to measure that. And this is given by WHO International Standard, which is the highest order reference material. And the use of the standard is mainly to define a common language to express the biological activity of a product. And this is international units. And there is in the right side here, there is a little vignette that I created in terms of vaccine and protective titer. So, for example, we all know that for hepatitis B, if you get your vaccine, 2-3 doses, then they test for your antibody, and if you hit the magic number of 10 international units per liter, you're protected. So, we ideally for SARS they want to do the same, but really, it's not that easy, especially with the new variants. And the thing is that you need to compare different studies, and you can only compare the study if we all talk the same language. Per se, the standard is nothing really that complicated. Most of the time, we are trying to have something that is very similar to a clinical sample. So for the antibody standard, for example, we just take convalescent plasma or serum from recovered patients. So there is nothing magical about the standard. The added value is the process in which the standard is evaluated. And this is very well defined by the WHO in the Technical Report Series 92. And it goes through all the stages. I've highlighted what for me is the most important step, which is the collaborative study. And I know in this room there are a lot of people that have participated in collaborative study, but for those that haven't, let me give you an example. And I picked the last fever example, mainly because this standard was developed in the middle of the pandemic, and I think we didn't have enough time to promote it. But it's a very important standard. Lassa virus is endemic in several regions in Western Africa, causing seasonal outbreak, and although majority of the people are asymptomatic, 1 in 5 infection results in severe disease, and the mortality for those that require hospitalization is actually quite high, 20% on average, while some outbreak can reach higher numbers. There is not licensed vaccine for this virus. And because of the potential of expand outside the geographic area, the WHO in Group 3, at least the one in 2018, had listed that as one of the top 10 pathogens for research and for development of vaccine in treatments. I think we had actually 3 cases in the UK, 1 reported cases, and that took us contact at the beginning of last year. So it is a real threat. So, sponsored by SAPI, we launched a collaborative study to develop this international standard and the reference panel, which is now available in our catalog. And they implemented this because they had at the time 6, now there are 4 vaccine candidates in their portfolio, and the 3 of those are currently in phase 1 clinical trial. So in this collaborative study, what we do is we send the candidate material together with other study sample. Everything is coded so people don't know what they're testing. And we ask participants, in this case, were 17 laboratories from 7 countries, to test the sample in 3 independent experiments. For Lassa, we received results from 35 methods. 16 of those were neutralization assay. Half of them were using the real virus, the other half were using outside the virus, either the lenti, a replicative VSV, or not replicative VSV. And then we had 18 binding assay. Of these, 13 were targeting the glycoprotein and 5 were targeting the glycoprotein. So the standard per se is, as I said, and nothing special, is well, it's very special to us, a while to find it, but is a pool of 6 donors from Nigeria, molested with plasma. It was obviously tested negative for Lassa virus, and this was kindly done for us by Mario Kirip at UKHSA. And then in-house, we further super detergent treated the sample to eliminate the presence of any other potential envelope virus. We also tested for the usual blood-borne pathogen markers like HCV, HIV, and hepatitis B. And we tested alongside this other sample. The reason why we have other samples is because the important things for international standard is one has to perform well in all the assays that are currently used, but the other one is we need to prove that the use of the standard that can harmonize the results of clinical sample for a laboratory that are using similar testing. And I have, as an example, only neutralization assay, but the same was true for the binding assay. So on your left, you have the data for each of the 12 samples, as reported by the participant in 50% neutralization titer. And the size of the boxes represent the spread data between the laboratories. When you reported the data relative to the candidate, which was a sample out of 4, you see how the spread becomes much narrow, and you have much more concordance on the results between the lab. So the data are normally statistically analyzed. There is all the statistical figures that show this increase in the interlaboratory variation through the use of the standard, and then we discussed the data at the expert committee on biological standardization, and they had established the standard for Lassa virus monoglobulin genes, because the convalescent plasma was from historical infection and was not EGM present in the standard. And then one of the issues we have with the standard, so this one, for example, we had activity for neutralizing antibody, 25 international units per ampoule, activity for anti-GP, and activity for anti-NP. What we find in the use of the standard is there is a misunderstanding on what this means. So these values are arbitrary. there is no correlation between the three of them. So there is not 10 times more anti-GP than anti-neutralizing antibody. For each one of them, we come up with a consensus from the assay used in the collaborative study to define the potency of the antibody, and they're not related between each other. Because of the frequent confusion, and nowadays the WHO has decided, the WHO has decided to split the antibody standard if there are different reactivity. For example, the recently developed anti-Griffin standard, there are two bios, they're exactly the same product inside. So if you want to go analyze an authorization message, just buy one of them. But they have two different labels to make 100% sure that people distinguish between neutralizing activity and binding activity. And there are other issues that we have encountered in the years with the international standard. I've come across people claiming that the assay was validated because the international standard was working well in the assay. You actually need a validation panel for that. People that would like to use the international standard every day in the assay, which would be lovely if we had a lot of them, but normally the stocks are quite low. And obviously the conversion factor, I have my NT50, well, actually the corresponding international unit. And that's this misunderstanding. International unit is an arbiter, is a kind of magical number that stays in there. And then in each of your assays will have your own NT50. So you can have the conversion factor that is work for everyone. And there are people that don't actually know how to calibrate the assay to the international standard. I've seen from very good laboratory the international standard being calibrated in the assay rather than the way around, and then claiming that doesn't work in that way. Surprise. So you cannot, for example, use your standard, the international standard 10, 20 times in your assay and come up with, oh, the international standard, my assay has an iteration title of this, so I can have a conversion factor. This is especially in cell based assay, there is always a certain variability. So you need to have an internal control in your assay, and that has to be calibrated to the international standard, and you report the results relative to that standard. And we have tried to help out in this through webinars organized by the WHO and with a manual that is available in the link on this slide. And the other issue we have found in the context of outbreaks is it takes a long time to make an international standard. They usually takes 2 to 3 years. I think we did very well for all the emerging viruses. We did about 1 year, and for SARS we managed in 9 months. But it's always too late. People have already set up the assay, and they're used to use the unitage they're currently using. So it's very difficult to move from that. So mindful of that, when COVID-19 came along, we decided to have a different approach. We provided people with a research reagent that was available towards the end of April 2020, which is high titer convalescent plasma for 1 UK recovered patient. This was possible thanks to the collaboration between ourselves, CEPI, and the NHSPT. And what we did during the next few months until the establishment of the national standard is to evaluate, to calibrate this research reagent to the international standard as part of the collaborative study, which has been mentioned here. So when the international standard was established, everyone that had used 2030, they could automatically convert their data in international unit by using the potency that has been assigned to the research reagent. And we did the same for monkeypox. Again, in collaboration with CEPI, we managed to have a research reagent available in November 2022. And I've organized a little pilot study with 4 laboratories involved, and the UKHC was part of it, BSD was part of that, and to evaluate this research reagent. And going back on the compliment, actually, this Medalli has published this paper in which they developed this neutralization assay, and they were evaluating how the use of complement approve the neutralization assay, so that was fine. And then we've been endorsed now by the WHO to move this research reagent to an international standard, which is the study is going to be launched this year. And then moving slightly from the vaccine and antibody to diagnostic. In the same way, we have research reagent that can be made available very quickly for antibody. We develop a similar strategy for diagnostic. This is kind of from the pseudotype field. So we can create a chimeric virus, which is an HIV particle, which contains the RNA of virus of interest. This is not replicative. We don't put any envelope, so it's not infectious. It's fast to produce, like you do a pseudotype, you just transfer two plasmid into nativity cells and becomes the UV agent. And the most important thing for this is apart from the fact that the RNA is more stable in a particle than naked, is actually function as a control for extraction. So it's treated in the same way you would treat a clinical sample. And the control for extraction is important in diagnostic because in several cases we have observed that there is, and that was true at least for Ebola, incompatibility from some kit and some amplification platform later on. And talking about Ebola, that's when we developed this strategy. So during the Ebola outbreak in 2013, the WHO asked us to produce an international standard for RNA, and we made two interim standards, which are the two products down the top right. And we use this technology because having the inactivated virus was taking a long time, and it's always difficult to prove inactivation, active, and we couldn't really take the risk to send out something that had any Ebola positive material. And the graphs on the left are from the collaborative study that was organized by Diana Wilkinson and NIBSC. So panel A is the results from one sample, and they are the copy numbers or not detectable unit for from several laboratories. There is a result 3-5 log. When you normalize data to the interim standard, you can see the majority of the laboratories actually achieve exactly the same results. One of the limits of this study was the lack, as mentioned, of comparison with an activated virus. With SARS, again, still remember it was January 2020 that my boss, Nicola, came in her office and said, shall we start preparing this reagent just in case? And so we did with Emma, and she created these particles with basically the 4 particles with overlapping section of the COVID-19 SARS-CoV-2 genome, and this is because of the size. You can only fit up to 9 kilobases in an antiviral particle. And we made this freely available to several laboratories, especially those doing diagnostic in UK. We asked for feedback, how much can you dilute the standard before you use Sino? And we noticed that the 2 laboratories using the same kit to amplify was an Altona kit, laboratory 2 and 5A, had 100-fold difference in their results. So we actually tried to investigate a little bit what the problem was, and at the end of several email exchange, laboratory 5A decided to change their extraction method, and they got exactly the same results as laboratory 2. So this is another example of how this is important, having this type of common research reagent to compare assay. And SARS-CoV-2 gave us the opportunity to test how this particle compare with the authentic virus. So when we launched the collaborative study for the international standard for SARS-CoV-2 RNA, we also include the chimeric lentiviral particle and inactivated viruses. And the results on the right, on the left, you have one sample of the results from all the laboratories that participated in the study. And again, there is a spread of results of 630-fold. And then in the 2 panels on the right, you have the results harmonized to either the inactivated virus or the chimeric lentiviral particle. Admittedly, the inactivated virus works a little bit better, But in both cases, you actually see this harmonization of the results, suggesting that at least as an interim standard in an outbreak, this is a very good strategy. And very briefly, we had the same results for glass RNA, which is a study that Emma conducted the year after, 2 years after. And we have again the harmonization achieved with the chemical lentiviral particle is very similar to the one achieved with the inactivated types. And I stop here. So there are a lot of reference reagents for high consequence viral pathogens. They are not all with NIBSC. There are 3 that mentioned custodian lab. One is the Paul Ehrlich and the other one is Cyber in the USA. And we are currently recruiting for some study. Jan is, I think, Mauro is recruiting one else, but still recruiting to Sudan, anyone interested. And Emma is currently recruiting for SARS-1 and 6th Sept, and I'm doing the MPOX, and I'm going to submit a questionnaire in the next few weeks. And I'll stop here. Oh, there's the acknowledgements. So there's a lot of collaborators. We couldn't all fit in one slide, so just put the one at MHRA and the funding body. That's it.